So thank you, Jesse, for the very nice introduction. And also Bob, Jesse, for this talk. I'm really excited to be here. I hope you can all hear me. Not too loud, not too low. Yeah, good. Okay, and if you have any problems with my accent, I'm German, so I mess up your nice English, right? So you're always welcome to ask me questions, right? I'm going to be talking about lichens today, and I have divided my talk in a couple of sections, and most importantly, I want to talk about what they are and why it matters. And so I want to start out with giving you background on the lichen symbiosis and how we understand that symbiosis today. That it's more than just a ménage à trois, and I will explain what I mean by that. And then I will talk a little bit about that these guys have been really overlooked for a long time. So I'm going to talk about them in the context of what's called the taxonomic bias in biodiversity sciences. And I will try to explain how that impacts how we understand their diversity. And I've worked for a long time in the Galapagos, about 10 years, and I'm still working in the Galapagos on trying to come up with an inventory of the species there and characterizing the lichens that are there and trying to understand how to better preserve and conserve them for eternity, hopefully, right? In that context, I will mention the consortium of Lichen Herbaria, which is a platform like you might have explored if you're interested in plants, SciNet. So it's a platform to manage lichen biodiversity data. And then I will switch towards Sonoran lichen diversity and the diversity of lichens here in Arizona. And in that context, I will talk about some of the projects we're doing here on the importance of lichens in the desert. And I will show you one of those 3D models, which I hope you like. Anyway, this guy, I guess you all know, which is the father of names of organisms, right? Linnaeus, he came up with this binomial system by which each species has like a first name and a surname. And he thought not much about lichens. He called them the rustici pauperimi, the poor peasants of the plant world. So this guy didn't really have a clue what lichens were, right? The first guy who kind of figured out what they might be was a doctor, a physician in Basel in Switzerland. And he gave a talk to the Basel Naturforschende Gesellschaft where he presented this idea. And he presented the idea that lichens were not just one organism, but two different ones. And he said in his talk that these lichens were, the gonidia and fibers of these lichens were not genetically related. Instead, the latter had to be a growth of fungal hyphae on algae. And Nylander was the expert in the field of lichens at the time, the world-renowned lichenologist. And Nylander called this a stultitia schwendenderiana, a stupidity of Schwendender. He thought this guy was an idiot. But we now know that he actually had quite a good understanding of what lichens were. So if you take a lichen on a rock like this one here, this is Xanthoria paritina, which grows, in that case, on a rock in the botanical garden of Zurich in Switzerland. You can cut it open and you look at it with an electron microscope, and what you will see that there are distinct layers built by the fungal hyphae. So all these threads are the spaghetti of the fungal hyphae, the fungus. And inside these distinct shape, there are round blobs, which are the algal cells, the gonidia, as they were called at Schwender's time, and which Schwender correctly recognized were different organisms from the fungus. What you can also do is you've taken a lichen spore, lichens like all other fungi are produced by spores, you throw that spore on another plate and you grow it, then you'd end up with something like this. Totally undifferentiated, like a heap of spaghetti that you take out of the water and dump into your plate, and there's no such structure. So the reason why the lichen grows in nature in a very organized matter, almost like a leaf, is that it is cohabitating with these other organisms, with these photobionts that make the lichen work essentially like a plant, even though the dominant organism in that relationship is the fungus. And here we have two general types of lichens. You can see these black ones, and then you see all kind of colorful other lichens. And if you cut them open, they look inside like this. So these guys here, the black ones, they are black because they contain cyanobacteria. And these chains here of pearls, there are the Cyanobacterium nostoc. And these guys here have green algae. And so these are quite organized, similar, at least analogous to a leaf. So they have typically an upper cortex, you have an algal layer, then they have a medulla, and they might have a lower cortex, right? And these cyanobacterial lichens are also organized in a similar fashion, but mostly they're a bit more chaotic, the cyanobacteria all throughout. Anyway, but I'm saying this becomes more complicated because there might be not just two partners, the so-called mycobiont, the fungus, plus the photobiont, the algae, but there might be a third partner present. And that was published only fairly recently in Science by a guy called Tobias Pribyl. And he did this. He looked at lichens with genetic tools, as we all do nowadays. We try to construct phylogenies of the organisms by using the molecular code, and we try to figure out how these organisms are related. But what he did then, he found in the DNA that he analyzed always some contaminations. So from the primary fungal DNA that he was interested in to construct his tree of fungal life, so to speak, he found a secondary fungus that didn't belong there from a totally different group. And he thought and scratched his head and thought, okay, why do I have always these contaminations? Maybe they are not contaminations, but maybe they're present regularly in those lichens. And what he did then, he basically knew that you can use fluorescent microscopy to target different organisms visually by different colors if you bind antigens that are specific to certain groups of organisms. So algae are photosynthetic, they're autofluorescent. Fungi, you can target with these antibodies specifically, basically bind these antibodies with some fluorescent dyes, and then you look at the things with a fluorescent microscope. So if you take a filamentous lichen like this and you cut it open, and you look at it with the fluorescent microscope, then what you see is three different colors of layers. So these ones are not green, but they're fluorescent violet, are the photobionts, green algae. And then he has these two different kinds of hyphae, the hyphae that are the thread like the noodle salad, right? And then they have some yeasts that are apparently, in this case, in the cortex. So yeasts, you know from bread, you know from if you drink beer or wine, are single-celled fungi, and you wouldn't expect them to be present as lichens. But when he did the DNA on these guys, he found that there are some of these yeasts in those lichens, and so there is, or seems to be, at least in the lichens that are mostly ascomycetes, so the typical lichen fungi, there are also some that have basidiomycetes yeast present. And that tells you that there are not just two partners, or not just three partners, but there are actually quite a few different partners present in lichen symbiosis. And it's very difficult actually to come up with a decent classification for them because you have two different domains. You have eukaryotes and bacteria. So the cyanobacteria are bacteria, photosynthetic bacteria. They are bacteria that kind of work like plants. Then you have the green algae that are algae, right? You have three different kingdoms. You have the fungi, so the green plants, the algae. You have the cyanobacteria. And you might even, in quite a few of those lichens, have two different divisions of fungi. So how do you put that in a box if you want to classify it? You can't. Well, for many years, what lichenologists have done is simply ignoring the fact that these things are so complex. They say, okay, well, we have one dominant fungus. It's the most diverse group. Yeah, so the algae are not actually as species diverse as the fungus. They're also what makes the lichen look like a lichen. So what you actually see on the rock growing out there is mostly the structure that the fungus builds up. the fungus dominates that relationship. It controls, for example, and suppresses sexual reproduction by the algae, et cetera, et cetera. So it's really the dominant partner. And so that means the tree of life, or lichens, is constructed essentially by the dominant fungal partner, by the so-called mycobiont. Yeah? And the first guy who came up with being interested in lichens rather than Linnaeus was Arcarius. So Arcarius is called the father of lichenology, and he came up with the first classification schema, and he looked at lichens, just how did they look like, what are all these shapes, and then he grouped them according to what they looked like. And that is what basically lichens look like. So you have crustose lichens. Crustose lichens are like paint. You kind of need to scratch them off, and it ends up with crumbles. And you need to basically collect them with their substrate because, like the paint, they are having an upper surface, but they're immediately attached to the rock or the bark or whatever substrate they grow on. And the foliose lichens, they have an upper side and they have a lower side. So they are leaf-like. So you could theoretically go underneath with the razor blade and pry them off their substrate, but often still you would collect them with their substrate because the substrate nicely supports them, right? And then you have fruticose lichens, and they are essentially three-dimensional. So they basically have branches. They might hang down, or they might point upwards. So these are the basic growth types of how lichens look like. But irony has it that we now know that the way they look like might not necessarily tell you how they're related, right? But it's all a bit more complicated. So the next guy who came up with this idea of how to classify lichens in a natural way, he thought, die natürlichen Pflanzenfamilien, the natural plants families, was this guy, was Saarbrücken. And he wrote the first big catalog of all lichens that were known, and we still these days use that catalog because it has all these wonderful names in it. And he classified lichens because he said, okay, forget about their growth form, look at how lichens reproduce, look at their sexual structures. And he was onto something because we know that reproduction really is a clue to evolution, so that was a much more natural system. So here's sexual reproduction. So if you take a lichen, that's a crustose lichen, you cut it open. So this is the so-called thallus, the body of the lichen. This is the fruiting body. Inside the fruiting body, there's an area called the hymenium. And in the hymenium, you find asci, which here are cut open. And you find in those asci, you find spores, ascospores. And so that's how these guys reproduce. And just to give you an idea, that the dominant part is really of the fungus. I have used this artificial coloring to color this from micrographs that I did as a grad student. And here are the photobiont cells, not very many biomass-wise in comparison to the dominant fungus, right? Many lichens, though, have a challenge. When they reproduce sexually, their spores land somewhere, they germinate, and they're very soon out of resources, and they're starved, right? So they need to find a photobiont to form a new lichen, because what lichen fungi do is they essentially feed on the photosynthesis, what the algae produces. So that gets into physiology of lichens. So we are heterotrophic organisms. Every morning you have breakfast, you have dinner, lunch, whatever, right? So you need to feed on something. Fungi also need to feed on something. You have a bread mold. Your bread mold eats your bread, yeah? And the fungus that you pick up in the forest and you make a nice meal out of it, that is not the fungus, but it's the fruiting body of the fungus, and the fungus actually grows inside the ground and feeds on whatever it can find in the ground, the organic matter. So these lichens, they feed on these algae. They don't kill them. They've found a way to cultivate them. So I always like to say lichens are like farmers, right? They cultivate algae. And now they have a challenge. When they distribute by spores, the spores land somewhere, the spores germinates, and the spore starves, unless it finds a photobiont. Now, here's another way to get around this. Distributing in little packets of hyphae, fungal hyphae wrapped around algal cells, or little miniature lichen outgrowths that break off, land somewhere, form a new lichen. Lichens also have, was when I started lichenology, that was the rage, trying to figure out, okay, what are all these pretty colors? Because lichens have a really very complex secondary chemistry. And there was a guy here in North America, or actually a couple, the Culbersons, and they analyzed all these secondary metabolites. And we now know that there's around 800 to 1,000 secondary metabolites, which the great majority of them are all exclusive to lichens and not found anywhere else. And you can use these different pathways and the production of these secondary metabolites to figure out how these lichens are related. So that's what the Culbersons then did. And now, of course, I've said we use molecular tools. And molecular tools have shown us actually a very interesting aspect of lichenization, namely that lichens have arisen in the fungal tree of life several times. So they have formed independently from one another. They're not a phylogenetic, close related group. Some groups, like the Leccanormycetes, are almost exclusively lichenized. But some groups, like the Basidiomycota, only have very few lichens. Yeah? So that means lichens are not necessarily closely related. They're basically a style or life form of how the fungi live. There's about an estimate of 20,000 to maybe up to 30,000 species in the world. And now I want to talk about something that I think it's great that you're all here, that there's so much interest in lichens, because that's not necessarily always the case. In the scientific community, people talk about this phenomenon of taxonomic bias. So when you and I go out, I might see a lichen on the rock, and you might not even notice it, right? You will see the big black bear that is hunting you, right? And you will see the beautiful butterfly, but you might not see the lichen. And so that results in that the way organism has received attention is very different. And for example, in the insects, this is about what we know, and this is about what we might not know, right? And the fungi, this is about what we know, and this is about what we might not know. And lichens, as I mentioned, are, well, for simplicity, lichenized fungi, and so they are somewhere in that column. And just to give you an example, when we studied some lichens in the Galapagos, there was this beautiful lichen called Cora. And it had one single name, it was Cora glabrata, and people assumed that it was all across South America. And we had a grad student who kind of scratched his head and said, well, I want to work on this molecularly and really figure out if that's the case. And that's what she did. And she then found that actually that single one lichen, what was believed to be a single species, was actually hundreds of different species. And so this is what she did. She went to, for example, the Galapagos. She went to many of these quadrants here that are, and these black ones are the ones that she sampled the most. And so she examined all these quadrants, and then with the other quadrants across South America, she compared, okay, how many species are these quadrants having in common? And from that, she extrapolated how many species there might actually be. So she was able, with her tools, to distinguish 126 species, and with that mathematics, to predict the biodiversity across the South American continent of 452 species, for what was previously believed to be a single species. And that gives you some idea of what kind of diversity is out there to be discovered. I worked, like I said, or like Jesse said in the introduction, for about 10 years in the Galapagos Islands, and I'm still working there, and I'm trying to publish like a flora or like a lichen book about the Galapagos. And I went to many different islands and many different sites, so about 500 sites. We collected about 15,000 specimens. And I looked at 10 herbaria, all the historic collections in the world, where previously lichens from the Galapagos had been deposited. This is Bill Weber. He was the first lichenologist ever to work in the archipelago, and he started in 1964. He got about 100 years old, died relatively recently, only 3 years ago. And in the Galapagos, we did something at the time. I was in charge also of managing the natural history collections on the islands. And we went across the different groups and looked at publications and what was there known in the collections. We tried to make a similar graphic, and we came up with this graphic here that illustrates the same kind of bias in the Galapagos organisms and we were able to estimate that around 80% of everything, of all the species in that prominent, well-known ecosystem like the Galapagos is still unknown. Yeah? So 80% of all species are likely still unknown. And just to give you an example of what we did in those couple of years that I spent in the Galapagos, Weber and later Elix McCarthy, they came up with a checklist of lichens of 228 records. Then now online there is a checklist with 792 records. And I have some unpublished records. So there's about 4 times more of what originally was known from the archipelago. And that means we found a couple of new records, new genera to science, new species to science. And we still have a long way to go. And here are some figures. So we started in 2007, or actually a couple of years before, but this is the publication of 2007, where we published about the genus Ramalina, found 50 species, 7 new records, 4 new species new to science. This is one of the more recent ones, 26 species, 30 new records, 10 species new to science. So that illustrates you taxonomic bias working on these kind of groups. This is a nice graphic that I really like by Stuart Peck. And he basically came up with an illustration in one book about the diversity in Galapagos, and he illustrated, okay, the size of these organisms. For example, the beetles stand for the insects. That is probably the species most diverse group in the islands. Here are the lichens, according to Weber. And this is what the lichens look like right now. So what I do is not just go out hunt lichens, but also try to characterize them. And so when I go out, I try to come up with some standardized way of collecting ecological profiles of these organisms. And there's a little paper slip that I carry in my pocket, and then I kind of quickly say, okay, they grow in a semi-shaded habitat on the front of a boulder in a wind and rain exposed northern side of that boulder, for example, right? And then I can analyze this data, and I can come up with some profiles, and I can say that species here, Roxella gracilis, typically grows on small branches. Well, excuse my French or my Spanish. That was done for a talk in South America. Anyway, so I can come up with these profiles for these lichens. So these are 2 endemic species of Uellia. And then going through the macro habitat, so the overall place where those guys grow, and the micro habitat, and I can find differences according to these ecological profiles that I can construct. And that is then useful in assessing the diversity of these lichens. And of course, together with the distribution of what we collect and where we collect it, and whether something is endemic, only known from the islands or not, we can then come up with some ideas of whether these lichens might be endangered or not. And here's a funny example that it took us a little while to figure out what was happening. So this is a cantolichen, this lichen, and it grows typically on these large trees, cinchona, in these closed canopies of an endemic scrub, Myconia robertsoniana. And these trees tower over these shrubs. They were introduced because of the bark. Cinchona is the quinine tree used to obtain quinine anti-malaria drug. And that quinine introduction was a mistake because now these trees are taking over that habitat. And so people in the national park want to get rid of these trees. But lo and behold, this species of lichen we now know is endemic to the islands. It only grows in the Galapagos. And it grows really and loves to grow on these trees. So we're thinking, okay, why is that? Turns out we found them also on a different volcano on a different island in this kind of environment. There's no trees there, right? But the lichen doesn't grow anywhere there. Where it grows is in these craters, and there are some of these tree ferns here. So it turns out this tree fern is a highly endangered, a critically endangered species in the Galapagos. It's also endemic to the islands. It only occurs in those islands. And funnily enough, we find this Galapagos spiny gladiator lichen, how we call the Galapagos, Acantholichen galapagoensis, on the fern fronds, on the sheets of those fern fronds. And we know that in the island where this canopy of Myconia has now subsequently been replaced by the Cinchona, that these trees, ferns, were very common and have disappeared. So they were probably the habitat of that lichen, overtowering the close canopy of the Myconia, which is very dark and shaded, and the lichens need some light, for obvious reasons. So they probably grew on those tree ferns. And because of that, because the tree fern is critically endangered, but the lichen is still there and it has found an alternative habitat, we then decided, okay, maybe we can classify this as vulnerable according to the IUCN criteria. So it was one of the first lichens that we actually was the first lichen that we ever listed at the IUCN as vulnerable. And this is how it looks like. And this is actually a page from the Consortium Lichen Herbaria. And the Consortium Lichen Herbaria is like a SciNet for lichens. If you, like I said before, if you work with plants, and you might have come across SciNet. So SciNet and Lichen Consortium, etc., they are all platforms built at Arizona State University upon a software called Symbiota. That's the software that drives these platforms, and is an extremely useful tool to manage collection information, create checklists, and manage the taxonomy of these organisms. And so here's the team, the Symbiota team. There's a lot of documentation, so if you're interested, you can go to Google Symbiota and learn about what this is all about. And like I said, it's a very useful tool. So in the Galapagos, I have used it to construct a checklist of lichenized fungi from the Galapagos. And then I've kind of had some success in trying to animate some of my students and some colleagues in Ecuador to collaborate with me. And we recently, and it's going to be published in September, I've been working on the checklist of all the lichens known from Ecuador, about 3,000 species. Now, well, 2,500, right? And 791 from the Galapagos, and basically still counting. And Alba and Angel and Rosa have been really the major collaborators in this. So now I'm going to do a U-turn towards the Sonoran Desert, because that's what you're probably all most interested in. I did my PhD like about 20 years ago at Arizona State under Professor Dr. Thomas Nash, who was the first editor and major author of these 3 volumes of the Sonoran Desert lichen flora, how we called it. So flora is maybe a no-no these days because we know that lichens are not plants, so people often then call this biota of lichens, but we don't want to split hairs here. And there's about almost 2,000 species known from that region, which means that it's about a third of the lichens known from North America. And so here's North America. That Esslinger checklist has about 5,000, getting closer to 6,000 now, species. The Sonoran region has about 2,000. In Arizona, we have about 1,000. And in Mexico, we have, only 2,700. That is probably very wrong. Again, that's another kind of taxonomic bias where people in these rich countries like this country have enough resources to work on these kind of bizarre things, whereas in countries like Mexico, they have some sort of other priorities, even though Mexico, like Ecuador, is one of those mega-diverse countries who has a huge diversity. So probably, I would estimate that in Mexico, they probably have at least 3 times the amount of species that we have in the US. So just to give you an idea what I'm talking about when I'm talking about the Sonoran Desert. So the Sonoran Desert is actually one of the very diverse ecosystems for lichens. And in the strict sense, the Sonoran Desert is this light gray here, which is in this post-Shreve map, these different bioregions of the desert. But the Sonoran Desert lichen flora, so these 3 books, cover a region that goes beyond that, so also these dark areas are included in those 3 books. And that's actually quite fortunate. We are one of the few regions that is actually fairly well investigated in North America. And just to give you an idea, Tom Nash worked on this for about his entire career with about 90 different authors for these 3 different books. Yeah. Anyway, so one reason why in Arizona we have such a high diversity of these organisms, these lichens, is the diversity, of course, of habitat. And you can easily illustrate that by going along an altitudinal gradient here across Arizona, right? And so just to start with something, because you have to start with something, right? Here we are in the lower desert shrub, and that is actually a habitat that's relatively poor in lichens. So, well, the saguaro is not really the ideal key species for that habitat, but it's more like these creosote bush. I would almost call them wastelands, because for lichens they're pretty boring. But these areas can be very interesting for some lichens. So here we have a species that grows on these desert pavements. And it not only grows on these desert pavements, but it grows in only parts of the rocks in those stones, inside the stones of caliche. So it's a so-called endolithic lichen. It's a lichen that grows inside rock. Here's a rock that you see. That's the pure caliche. That's where it's been infested by a lichen. So these round black holes in the rock are the openings of the fruiting bodies of that lichen. So if you cut this open and you section it and then you stain it with a special dye and you make sure you preserve the organic matter in it, it's a bit complicated. I can talk about it in more detail later if you like. Then you can see that this lichen has grown through the entire rock. And this is how it looks like also with use some other techniques. There's a micrite layer, so a little layer of newly formed limestone on the top. Then there's this algal layer stained black by a color called, or by a stain called osmic tetroxide, otherwise these would be green. Then you have this pseudomedulla, so where the hyphae actively grow into the rock, and then you have this lower layer where the rock hasn't been penetrated yet. So I did this quite a while ago with a geologist, and you might wonder, okay, yeah, they can grow in that caliche, they're amazing, they even built their little greenhouse, but what about water? So lichens are really fascinating in another regard. They are so-called poikilohydric organisms. So you and I, we are all homothermic, and the lizard is poikilothermic, which means the lizard can't heat itself. It constantly has fluctuating body temperature. It needs to go in the sun when it's cold, and the shade when it's too hot. We regulate our own body temperature. So now if you take that term and you translate it into hydric, then poikilohydric means that these organisms are not able to control their water content. So they don't. They basically completely dry out. When they're dried out, they're not dead, they don't die, but they're dormant. They shut down their physiology. And then once there's water around, they resurrect. They have a series of enzymes that rebuilds the cells and reconstructs the cells and makes the guys live again. And here's an interesting experiment from that botanic garden in Zurich. Rosemary Honecker put some lichens on a plate to examine them in an electron microscope in the absolute vacuum. Even put some gold layer on top of them to see what their structure looked like. Then she thought, okay, what are we going to do with that stuff? Oh, I'm just going to put it out in the garden again and see what happens. Lo and behold, this stuff would go back into a nice living lichen. Because when the lichen is dry, you can put it in an absolute vacuum, you can put it in nitrous nitrogen, you can shoot it out to outer space, and the lichen says, Houston, we don't have a problem. Yeah? So these guys are pretty hardy when they're dried. When they're wet, that's another thing. You can instantly kill it by throwing it in a vacuum or in liquid nitrogen when the lichen is fully hydrated. Yeah? So back to the Sonoran Desert. From the lower desert, we go once up to the Superstition Mountains, so the higher desert, and there you will see that the lichens are sometimes covering entire rock surfaces, sometimes even the entire cliffs are painted bright yellow, and you might notice this on your next hike. And we have been trying to understand, okay, how do lichens affect rock surfaces? And there's lots of investigations how they actually dig into the rock and what they do, how they affect rocks. One of our studies I've shown you. But we know very little about the time dynamics of this process. So we're trying to monitor lichen growth on rocks. And you could go there with an acetate sheet and you just trace them, and then you go back the next time and trace them again. That's very inaccurate. So what we've been trying to do is we have tried to use a new technology that mostly ecologists and some game designers actually use for virtualizing stuff in 3D by using two-dimensional photographs. How is that going to work? Well, if you have a camera and you move the camera around and you take pictures at different angles, then you basically have pixels that are the same in those photos and they overlap. And from those overlapping pixels, you can throw that into software that will then analyze the photos and create what's called a cloud of shared pixels. And it overlays a network, a mesh, on top of that cloud, and is able to reconstruct a 3D model from these pictures. So we have these rows of images that are all overlapping at different angles, looking at the surface, and then we throw them together and make a 3D model. And so basically this is our 3D model, and this is the mesh that lies under this 3D model. And now I want to play that video, and I have my brave assistant here who is going to do that for me because I'm not used to Mac, so just to give you an idea what that actually looks like in reality. So now you can imagine yourself as being a little ant moving across the rock surface of lichens. So that grad student that I have, that's the video, Guillermo Ortiz, he has been trying to wreck his brain by figuring out the standardized method that really works, where we can take the same shots of the same areas over time and use that for monitoring purposes. And as you can imagine, there's very many technical details that need to be figured out. One big challenge that you face when you do lichen macro photography is that you run into problems with what's called depth of field. In photography, when you do macro photography, you never get everything sharp. And the only way to do that is by having all these images. And so these single images that are in full focus, they're actually, each one of them a stack of images at different focal planes, and they're being mounted together. So a model like this has 2 rows of images. Each of these images has to be overlapping by 60 to 80%. Then they're angled and plane photos. Yeah, so you look at the structure of motion thing, right? You have to have different angles. And then each single image is actually a stack of 30 photos. And so if you all multiply that, you end up with about 3,000 or something photos for a model like something like this, right? Okay, another turn. I can't do everything by myself, of course. And it was really fortunate that there are people in Arizona who are interested in lichens. Right, and one of them is Gary Neal, and he is a volunteer for the Red Rock Ranger District of the Forest Service, working with Janie Agayos, who's the wildlife biologist there, and he is an avid lichen hunter. So he basically goes out there, looks for lichens, doesn't just take pretty pictures, but brings them home and tries to figure out what they are. And he had to, in his garage, set up his little lab to do that with his microscopes. Unfortunately, you can't just identify lichens by the look of them. What they look like from the outside might be quite deceiving. You need to often cut them open, even do some chemical analysis, et cetera, et cetera. So they're on this borderline of being rather difficult to identify just by their looks of them, right? And in Sedona, in that area, there's also a habitat that's still pretty unique. And that's maybe amongst the most threatened habitats in the Southwest, and those are soil crusts. So why are these soil crusts so threatened? So soil crusts are basically lichens are not good competitors. So they are competing with other phototrophic organisms, namely plants. And plants are much better competitors. But in a climate like this one here, where it's really dry, in this arid southwest, plants have a hard time. Yes, the big columnar cactus stores water, but if it doesn't get water, it shrivels up and dies. And the lichens, as we say, they can survive long periods of being dried up and they don't bother. And so in the natural soils of the desert southwest, all these blank open soils would normally be covered with these guys, with soil crusts. And soil crusts are actually very, very complex ecosystems. They're not just lichens. What you see here, the bright color and the black color, those are, to the naked eye, they're mostly lichens. But if you cut a cube like this out of the soil, you will see there's other organisms there. There's cyanobacteria, there are fungi, there are green algae, there are lichens, there are bryophytes. So it's a very complex ecosystem. And if you tread on the soil crust like this, it might take another 200 years until it's back to that beauty. And so Janie has taken a particular interest in protecting these guys. So if you go have a look at their Facebook page, there's a nice video about soil crust where she explains the importance of these. And, well, I don't have too much time, but I want to give you a bit of everything of Arizona. And so one of the really interesting things, of course, in the desert is that out of these the desert islands, and out of the Colorado Plateau particularly sticks a huge mountain called the San Francisco Peaks. And one of these peaks, Mount Agassiz, named after curator, plant curator Agassiz at Harvard, is off limits. So you go up there, you can take a lift, you can look around on that platform, and then you can take the lift down, but you can't walk up on the mountain. And the reason for that is because on that peak there is the San Francisco Peaks ragwort, which is a plant that only grows on that single peak in the world, right? And so you don't want to have people walk on that mountain and do this on the Arizona ragwort, because then maybe you won't have it growing anymore there, right? And interestingly enough, we also went up there because we had a special permit. And we didn't pay the $500 fine, but we were looking for this guy. This is Lecanora subcavicula, and we're trying to figure out whether this is the same species as on some of the peaks in Nevada, or whether it's a different species. So it might be that this lichen here, Lecanora subcavicula, described from the San Francisco Peak, is as rare and as unique as that ragweed. Yeah? So there are some lichen endemics, but they're a bit more sensational because, as you heard, lichens get around by spores. They're part of the air plankton. They're being blown about. So the general assumption often is that they're not as unique to a place, and they're more widely distributed. But some species in the Galapagos, here on the San Francisco Peaks, are probably unique to these sites, and they're probably endemic. A really interesting area that I didn't want to leave out of the talk, because it's probably the most biodiverse part for lichens in the Sonoran region, is Baja California. Yeah? And Baja California is unique so far because it's a fog desert, especially the side on the Pacific side. Yeah? So if you ever camped in the Bahia San Quintin and you get out of your tent in the morning, you wonder why it has rained in your tent. Because in your tent there's dewdrops everywhere. And you just open your tent, you look out, and you see that in the bay there's a big fog bank that is slowly disappearing in the morning. And so that's the reason why it's so humid in these areas. So it's a very dry desert, but you got the fog and the dew, and that's what the lichens thrive on. Yeah? So these Fuchsia columnaris trees, they look like Alice in Wonderland, the bosom trees, like a stick of a carrot sticking out of the desert, are draped in these lichens, Ramalina menziesii, the lace lichen, which actually is perfectly adapted to capturing this fog because it has these branches that are open like lace and where the water can condense. Now this is an interesting lichen This here on the left is Polycalyona thamnodes from Baja California. And this here is Volmania orthoclada from the Atacama Desert. The Atacama Desert is also a fog desert. Also the fog is rolling in there. It's even a more extreme desert actually than Baja California. And these two lichens we now know, although they look very similar, are not related, or at least not closely related. They're in the same family, but in very different branches of the family tree, so to speak. And I've talked about that lichens are crustose, foliose, and fruticose. So this guy is a crust, right? But it also has these funky outgrowths. And this guy is a crust, and it also has the outgrowths. In fact, in some areas it looks almost entirely fruticose. And that shows you that under certain environmental variation, it might be beneficial for a lichen to grow even in different ways, right? And so that's why sometimes the look can be quite deceiving. And our hypothesis in this case is that the more humid it is, the more the lichen needs to kind of get rid of water, sort of a thing. So it kind of stands up and forms these three-dimensional growth, which will more easily get rid of the water. And the drier it is, the more it can afford to grow or keep growing as a crust. And the people who have done mostly that research is not myself, but this is Ronaldo Vargas and Daniel Stanton. And here they're growing, here they're not growing, here they're working in the Atacama Desert. So that's the Atacama Desert. That's a desert that receives, on average, rainfall every 10 years. Yeah? But it's very diverse in lichens. So even though you look at these rock-strewn slopes here, if you look close, there's lichens everywhere. And again, the reason is because the fog is rolling in every morning and rolling out during the day. And so the lichens, they go through this resurrection life cycle on a diurnal cycle. What they, by the way, do in the Sonoran Desert as well, here in the early morning when it's humid and shaded and not quite as hot, they are active, they're photosynthesizing. During the day, they're dormant. At the evening, they start becoming alive again, and now they have a problem. During the night, the fungus is a heterotroph. It feeds on something. During the night, it's humid. The fungus is alive. So it sucks all these photosynthetes from the algae. And then in the morning, you have that short window where the algae can be productive. So now, if climate changes and that window where the algae can be productive shrinks, then these organisms, even though they're so hardy, might have a problem. Because then the algae might not produce enough photosynthesis for the fungus to have a meal when they're even not productive during the night, right? So it's all a balance. So these lichens, they are quite amazing. They can survive very extreme conditions. They can even live in a place like the Atacama Desert, but they can't survive everything. And in fact, if you go into the desert, you will easily see that. You can look at different sides of a boulder. On the northern side, often you find an abundance of lichens, whereas the southern side of a boulder might be almost devoid of any lichen growth. And with that, I conclude my talk, and I'm open to receiving questions. Thank you. And, well, before you clap, there's, of course, lots of people that I want to thank, not last but least, the National History Institute here, and then people at the Biodiversity Information Knowledge Center, Bosch Thompson Arboretum, there's the Lichen Herbarium, the Galapagos, et cetera, et cetera, et cetera. So I will stop here. You can read it yourself.
Hooray, thank you so much. Also, I found this giant lichen book in the NHI library if anyone wants to look at pictures afterward. I don't know, is this a good one?
This has spectacular pictures of
spectacular
lichens.
Pictures. So we have time for a few questions. And then afterward, we brought down NHI's limited lichen collection, and then we have a few others that Frank brought up from ASU. So, oh, and Frank, if you don't mind repeating the question into the mic so the folks watching at home can hear it. Thank you.
Simple question. When we did geology in Montana, they were saying it looks like
Yeah, so the question, I guess, is in Montana, when you looked at rocks, that there are different species on calcareous rocks, on limestone versus siliceous rocks, so things like granite, basalt, whatever. Yeah? And the answer is you have very good eyes, because lichens really, like vascular plants, are quite different according to what substrates they grow on. So in vascular plants, you have calcifieds, plants that prefer soils that have a lot of calcite, whereas some that don't live on these calcareous soils. And with lichens, you have the same. The lichens prefer to grow either one of those rocks, and they can be very specific. Some, in fact, they can even tolerate high copper content by making this inert by producing mullite, an oxalate of copper. So, and because these lichen species that are growing on different rocks, these species also have different colors. And yes, many of the particularly colorful ones, the bright orange, bright yellow ones, often are found on calcareous rocks, but not exclusively. Are you oh, sorry, maybe you were first.
What is the effect of the change in the lighting within the colony? All other things being equal, do they just slow down in their...
So the effect is what, the question is what, yeah.
Built as a sunshade over the rock that has the lichen on it.
Yes.
The rest
So,
of the environment is the same, but not as much light.
right. So the question here is about the light. What role does the light play for lichens? And it has a huge impact on what kind of lichens you find on rocks. So there are species that prefer to grow in the shade, and there are species that are adapted to tolerate high light input. And these pretty colors are actually sunscreens often, not all of them, but these secondary metabolites that are so diverse in lichens often act as sunscreen. And there's a team from Columbia doing research on that, and they have proven in laboratory tests that many of these secondary metabolites from lichens are even more effective than some of the chemical sunscreens that we currently use in sun lotions. And it's difficult to commercialize this because there's nobody who's figured out a way how to grow enough quantities of lichens by growing them quickly. So yes, light has a very high impact on what, but there's different species with different preferences.
What role are the symbiosis yeast playing? Are they living on byproducts, or are they contributing in some way to the lichen itself?
Yeah, that's a very good question. So the question was, what role do the Basidiomycete yeast play in the lichen symbiosis? And the answer is, we don't know, but we have a clue. So first of all, this idea of being more organisms present in the lichen symbiosis is a relatively new one. And we don't actually quite understand if that's present in all lichens or only in the few ones that have so far been studied. Yeah? In that particular example that I showed, those yeast were mostly found in the cortex. And so there's some speculation, which people haven't tested yet, that when you try to synthesize or resynthesize a lichen in the lab, in the laboratory, by bringing the two partners back together, the mycobiont plus the photobiont, then these experiments have largely failed. And the general assumption was always, okay, the conditions in the lab, you can't mimic the habitat in the environment, and therefore these experiments failed. But another idea that these guys working on these yeasts have is that there's a structural component missing, namely the yeasts that are maybe helping to hold the cortex together. So that is one idea. But actually, lichens are not just these few organisms, but we now know that there's other organisms present in lichens too, so endolithic fungi. So you might have heard about endophytic fungi, so fungi that live inside leaves. Yeah? And nobody exactly knows what their role is. And you have found these fungi also growing inside lichens. What is longer known than endolithic fungi are so-called lichenicous fungi, fungi that parasitize lichens. So they're like an additional partner that sometimes is like a disease and causes damage to the lichen. But some are probably more like parasymbionts, kind of cohabitants living in the lichen symbiosis. So you have your typical fruiting body on the lichen, then you find another fruiting body of something else. And that shows you that there's other fungi present. And so you could make this argument that lichens are, in a sense, micro-ecosystems of all kinds of different organisms in that little scale that you're looking at. And the role of many of those things hasn't been quite figured out yet. Oh, you have a...
Go ahead, Jenny.
The lichen breaking down the rocks.
So this is a complicated story, and I didn't want to delve too much into this, because they do a little bit of both. So in this example of the Araucaria or Bagliatua rubucincta that we examined when I was a grad student with this geologist, we found that there's evidence that this little calcite layer on top of that lichen is formed de novo, meaning it's not there if lichen isn't there. And you can also see when you look at these rocks that you think the lichen would kind of grow into the rock, and then the rock would look something like this where the lichen is. But it looks something like this. So that means the surface of the lichen is actually producing, in a way, that additional calcite layer on top. Meanwhile, the lichen hyphae are digging actively into the rock and disintegrating the rock. And so this is probably not an active production where the cells are producing new calcite, but it's an indirect factor of what's happening there. Remember, you have two different organisms. The fungus, for example, excretes CO2. It breathes out CO2 like we do. In aqueous solution, CO2 is a weak acid, yeah? And that weak acid, your carbonic acid that you have when you have a sparkling bottle of water, is released, right? That makes the sparkles. But in the rock, it can deteriorate calcite. Yeah? Also, the lichen fungus produces oxalic acid, which is a stronger acid. And you can find calcium oxalate down there in the rock. But the algae does something the opposite. It's a photosynthetic organism. So it binds out of the environment, it sucks CO2 out, and it releases oxygen. And so at the surface where the algae is, or above the algae, probably the reverse process is happening, so that like in a stalactite cave, where the stalactites form when water evaporates, then because of that reverse process, probably that surface layer is accumulating. And so literally the lichen is most likely, there's a bit of speculation, essentially building its own greenhouse by digging into the rock and producing a roof on the top, right?
Wow.
And I mean, that's just one story, but essentially it becomes really complicated because all the lichens do something a little bit different. And so some lichens might probably be rather damaging for rock substrates and deteriorating rocks and initiating soil formation. And other species might form a protective layer that, even though they kind of dig into the rock and they do some damage, also holds this whole stuff together. So it is a rather complicated story, and people haven't really figured out the ends of it.
I have a question from the live chat. Maria wants to know what kinds of lichens glow at night.
Very good question. So there are actually quite a few.
Yeah.
So when you look at all those pretty colors, then you can analyze these colors. You can basically do a technique called thin layer chromatography. You throw the lichen into acetone, you extract these molecules, you put them on a thin layer plate, so a plate covered in silica gel, you move a solvent through them, you discriminate the different substances by their moving on the plate, but you also put the plate under natural light. You look what the color is under natural light, and you can put it under UV light. And some of these lichen secondary metabolites fluoresce. And so if you go out in the desert with a flashlight, UV flashlight in the night, and you light the rocks with your flashlight, you will see and notice that some lichens brightly fluoresce because they have these secondary metabolites that do that fluorescence, cause that fluorescence. And there's no general group that does fluoresce and another one that doesn't, but some species, depending on what secondary metabolites they contain, do fluoresce and others don't. Sorry?
When in the geologic record did the
Oh.
lichens?
That's another really good question because there's very poor fossil records for lichens. As you can imagine, lichens don't preserve really well, and also they're mostly microscopic, and so some of the structures that we have in the fossil record are interpreted as lichens, but there's a little bit of doubt. So there's good evidence from the Devonian that people are pretty convinced they're lichens in amber, right? And so we do know that they date back quite a while. And the general assumption nowadays, and there's some views challenging that, is they probably came on land somewhere around the same time as the early land plants. And some recent publications make an argument by dating the molecular clock that probably they arrived a little later, right? But it's all very iffy because, like I said, the fossil record is really poor.
I think let's do one more question, and then hopefully maybe Frank will stick around and take whatever other questions. I know Ellen has some questions, some other questions afterward. So Russ, if you...
Okay. How do you know when you have a new species? We're talking about a community of plants. At least that's kind of the way I would phrase it.
Mm-hmm.
And that would be a continuum, I would think, rather than distinct orders. So it seems like when you draw a line and say this is a species,
Mm-hmm.
or this is in between species, or am I just looking at it completely wrong?
No, you're not looking at it completely wrong. You can actually discuss forever with scientists what a species is. And it's very difficult to answer where to draw the line, essentially. So with lichens, like I said, you have this crutch of saying, okay, I call this by the dominant fungus. That's what I give a name to, right? And you ignore all this other stuff that I talked about, that they're actually microecosystems. So you name the microecosystem for the dominant component. But then again, it's also tricky with that dominant component to figure out which species you're dealing with. And traditionally, like I showed you, the first guy, the father of lichenology, Acarius, tried to look at lichens. Okay, this one looks different from that one, so that must be a different species from that one. And to a certain extent, that is still going on. It's more refined now. You're not just looking at it macroscopically. You're also doing the microscopy. You look at the anatomy of the spores, the alter structure. You look at the biochemistry of these things. And you more and more use, and now almost always use, molecular tools to figure out how things are related. But even then, if you end up with a tree with all these branches, where you cut the tree and say you call that a species is sometimes in the view of the beholder. Basically, you make a decision as a scientist and a species, and that's why taxonomy, naming things according to how they are related, is a science, because it's a scientific endeavor of erecting a hypothesis. You name this, I call this species, and that's my idea of what I think this is, right? And I have good evidence, and I make a long description, and I publish these results, and then another scientist comes along and says, well, hold on a minute, I don't think so. And so, but that's the scientific endeavor. And you, basically, as humans, what do? You have to deal with these boxes. You need to have something. If you go out and you call this car a Ford and this car a Chrysler, that's easy because they're produced in different factories, right, by different companies. But then in the Ford Chryslers, how do you split them, right? I mean, it's kind of, you can say, where do I draw the line? But at the end of the day, you always have to have something where, if you're an ecologist, you want to go out and say, okay, this is happening here, and I need to give these things a name. Otherwise, I can't deal with them. Yeah.
Some of them have your name, I guess.
Well, that's attached to, so basically, when you name an organism, you have your name slapped at the end, often because sometimes what happens is that people give the same, give different organisms the same name. So that's a so-called homonym. And in order to be able to distinguish how people named them, you have to have the author of that name. And then in taxonomy, you have the rule of priority that the oldest name, the one that came first, has the right of priority, and that is the name that the scientific community accepts. And your other name goes out of the window. And so if I describe a new species tomorrow and somebody comes along and proves me wrong, then that's bad luck.
So one more round of applause, everyone, for Dr. Frank Fungi. Yay! Here we go. As a thank you from the Natural History Institute, we picked out this special lichen-colored hat. Just for you. There you are. Thank you so much.
Okay.
And if you all have extra questions, you're welcome to come and chat afterward. So, and the gallery's open, so thanks all for coming.